presented?
It's okay.
That's okay. Okay. Today we'll speak about fundamentals of biological age. We will speak about this concept and why this concept is important, especially for aging domain today. Before we go to the biological age, let me first explain what is surrogate endpoint. Surrogate endpoint is a measure which went from the clinical trials domain that measure an effect of a specific treatment that may correlate with real clinical endpoint, but does not necessarily have a guaranteed relationship. For example, surrogate endpoint can be convenient for clinicians because the true outcome, like the death, is undesirable outcome for clinicians, and we want to somehow measure some value which can predict the undesirable outcome before it occurs with individual. For example, cholesterol level could be a surrogate measure for cardiovascular disease. We may just measure cholesterol level to understand how close a particular individual to the disease. So it can be used instead of a real outcome, which is undesirable, of course. However, sometimes surrogate measure may lead to wrong conclusions and lead to developing the drugs which are inefficient, which are may lead us to the wrong conclusions in clinical trials, and thus surrogate endpoint is an object of a critics also. Why I started from the surrogate endpoint? Because we need some definition of biological age. And here's the problem. There is no unique definition of biological age. I proposed 3 definitions here. The first one is quite maybe the most common and quite old, that biological age is a quantity expressing the true global state or organismal health state during aging process. And it reflects this state better than corresponding chronological age of an organism. The second definition is not a definition actually, but I proposed it here because it was mentioned in quite respectful paper from Nature Review Genetics journal for this not definition, but kind of synonyms of biological age, which can be also treated as physiological age, organismal phenotypic age. And Professor Horvath just emphasized in this paper that this definition is quite ambiguous and actually doesn't have a precise definition. But finally, I found one interesting paper from clinicians, quite recent paper from clinicians, where biological age was proposed as a surrogate endpoint by definition, reflecting health state of an organism. And I think this is quite a good definition because this allows us to formulate criteria to a good biological age. The first criterion, biological age should correlate with a population mortality, because we want to predict undesirable outcome, as I said before. Mortality or death of individual is an undesirable outcome, and biological age in principle should be a predictor of mortality, of a death. The second criteria, at least biological age should be capable to predict aging-related disease, cardiovascular, cancer disease, diabetes, and other chronic disease like fibrosis and so on. And the third criterion is this quite optional criterion, but I believe quite meaningful, that biological age should correlate with perceived age, or also known as self-rated age of an individual. So these three criteria are quite transparent. And the last definition we need today is a definition of aging clocks, which is very simple actually. Aging clock is just an algorithm predicting biological age. Now we are moving to the mathematical domain, slightly and slowly. This formula shows the typical assumption about biological age model. We typically say that our true biological age is decomposable to the chronological age and plus some delta, which is called age acceleration term. Chronological age is obviously an observable value. We know our chronological age, but age acceleration, not. Age acceleration cannot be measured directly. We may just assume what should be the age acceleration. For predicting biological age, we're typically using aging clocks. Yep, aging clocks is a function that computes an estimate of biological age from a set of biomarkers that can be blood parameters, DNA methylation, metabolism, gene expression levels, and it's depicted by function f here. Aging clocks f predicts we say biological age from biomarkers, and this aging clocks model will be random forest algorithm, multiple linear regression, MLR, support vector regressive neural network, any you may imagine. This approach to biological age estimate generates one problem, namely how to distinct noise from true biological age acceleration signal. What does it mean? This biological age estimate is a part of true biological age. The another part is an epsilon term, which is just a random noise. If we substitute the formula for biological age, main assumption here, to the bottom formula, you see that biological age estimate can be rewritten like this. This is a chronological HC plus delta minus epsilon. And these two terms, they are actually random terms. And the problem is that they are indistinguishable in practice. We cannot distinguish the true biological signal from the technical random noise in this formula. So our estimate suffers from this indistinguishability. The second problem is a specific problem for supervised machine learning algorithms trying to predict biological age. The problem is formulated as a biomarker paradox, namely, what if there exists a biomarker predicting chronological age ideally? This means that suppose we have a biomarker X which predicts biological age exactly as chronological age. Suppose that such biomarker exists actually. So in this case, there is no delta term because B is precisely equal to C, and such aging clocks becomes useless because we don't have delta term here. The third problem is how to use chronological age as a feature for biological age prediction. This problem is also specific for supervised models, which cannot use chronological age for biological age prediction because they use chronological age as a target for training. Although it is quite natural to use chronological age as a proxy for biological age. Why we can't use it as a proxy? Because it's quite natural. We know our chronological age and we need to just predict this delta. To overcome problems 2 and 3 and partially the problem 1, two general approaches exist: unsupervised approach and biological age prior model assumptions. Today we will not speak about unsupervised approaches, but there exist two. For example, PCA-based, principal component analysis-based age predictors. But today we will speak about model assumption, about approach with model assumption. Namely, we will speak about Klemera Doubal method. Okay, Klemera Doubal method. We're starting from the assumptions of Klemera Doubal biological age model. The first assumption is quite general. Aging is a natural process which is manifested in change of many properties of living organisms. The speed of the process is markedly different for individuals of the same species. This is quite general. It is assumption which you can see in many papers. In principle, most of the authors agree around this assumption about aging. The second assumption we already seen, but they, Klemera and Doubal, add an important statistical piece in the assumption, namely the expectation of biological age in cross-section population data is equal to the chronological age of this cross-section. Consequently, the expectation of delta equals to 0. This is quite interesting assumption, but it is also quite meaningful, I believe. The third assumption is any measurable property of human organism that changes systematically with chronological age might be affected by the individual degree of aging and used then as biomarker. In principle, I do not see any problem with this assumption because it's quite also natural to assume that there exist biomarkers which are affected by individual degree of aging. And they can reflect this individual degree of aging. Okay, the last assumption is very important here. Very important for this particular paper because it poses that any age-related biomarker can be functionally represented through biological age and biological age independent fluctuations delta x. Do you remember the problem 1, by the way? The problem 1, we considered earlier. I just remind that problem 1 is how to distinguish noise from true biological age acceleration signal. This assumption they propose here, it is an attempt to introduce this noise explicitly to the model of biomarker X. Authors understand that age-independent random noise exists, and they include it explicitly. And this is very meaningful too. So the first ingredient we need for understanding Klemera Doubal method is univariate linear regression. Let me introduce such denotation, y equals to c. When we will speak about y, we will speak in most cases about chronological age because it's using it for supervised model training. And we need, yes, as I said, the first ingredient is univariate linear regression formula. When we try to predict chronological age y from the biomarker value x. Klemera and Doubal use inverse linear regression, trying to predict biomarker value x from chronological age y with some weights, with some slope, and some intercept coefficients. The second ingredient we need is follows from higher school, I believe. Maybe you have seen this in your higher school. It is the parameterized curve. parameterized curve that can be defined by a single parameter t, for example here, which are functionally coupled with coordinates in plane x and y. Here we may see a parameterized circle where different values of parameter t determines a particular point of this circle. Analogously, you may, for example, determine curve in this space. For example, in 3D space, you may add additional coordinate z and determine a spiral function, spiral trajectory. Now, suppose we determined m linear equations of the same parameter y. These are equations for all our biomarkers in a particular data set. My question to you now, what will be the shape of the curve in multidimensional space? Try to think about this. It's quite interesting. Guys, are you here?
Yeah, I'm thinking.
Yeah. Who was that?
Oh, just me, Dylan.
Can you imagine the shape of the curve that is determined by this set of equations? Just the shape. Just, yeah, it is a multidimensional curve, of course, but maybe you can imagine the shape.
Must be the same y. They go in opposite direction? No. It's been a it's been a while. My mathematical brain is not quite.
Yes, probably it's not. Yes, maybe sorry, I slightly biased because I had a lot of experience in my multidimensional in mathematics of higher dimensionality, but actually the curve which is parameterized is just linear, but linear embedded in multidimensional space from x1 to xm, and these axes is kind of orthogonal coordinates in the multidimensional space. This is just a straight line, and the points, the samples of our data set are spread around this straight line in m-dimensional space. And this line determines the kind of best guess values of these points in the multidimensional space. Yeah. Now, suppose sorry, then yes, the next part. Now suppose that we obtained a new point x new from the test dataset. We didn't see this point before. We obtained a new point from test dataset, and the question is, what value of y new does it correspond to? It turns out that we can reformulate this problem of finding the best value of y in the following way. Assuming that our points are embedded in the Euclidean space, we may determine this best of y cap, as just such y that minimizes the distance from x nu to this blue line, this blue parameterized trajectory. So, and what is quite, I would say, tremendous, that this particular problem has a closed form solution. This solution is called Klemera Doubal solution, or unweighted Klemera Doubal. Why unweighted? Because different biomarkers have different levels of association with age, with chronological age. Some may have a high correlation with chronological age, others a poor correlation. And we would like somehow weigh them to account these differences in biomarkers. How we can introduce weights? We may just to add weights alphas into the formula for computing the distance for computing the point y cap. And Klemera Doubal in their paper showed that the best choice for alpha is the inverse residual standard error. I just remind that residual standard error is just a variance around a linear model, how points spread around this straight linear line. This variance, of course, is big for this particular linear model, and this variance is small for this particular linear model. Substituting this residual squared error to the formula and recalculating the whole thing gives us weighted Klemera Doubal formula. And the last ingredient we want to introduce, we want to understand, is the introduction of chronological age into the formula for biological age estimate. Yes, I did not mention this, but this formula is already for biological age estimation. Y cap nu is a biological age estimation here. And to introduce chronological age into the formula, we need to use an assumption 2 from the Klemera Doubal assumptions. The assumption 2 poses that C equals to B plus some delta. This means that C equals to B plus some random noise, which is biological noise, of course, biological fluctuations around chronological value. Substituting, we may invent additional equation which express our chronological age as a parameter y. But as you remember, the chronological age is in principle is equal to the c as we determined it before. This may be quite confusing here, but probably it is because my denotation may be quite confusing here. But the idea is that we may parameterize chronological age within our model just by using it with just by using this equation, c equals to y. This means that c equals to y with the slope equals to 1 and 0 intercept in the linear model. And substituting this to the formula and recalculating all this stuff, we may obtain the final biological age estimate formula, exactly how it was proposed in Klemera Doubal paper. However, note that this term has a strange term SB, which is equal to standard error of biological age or variance of biological age. If you are interested how it's computed, I will answer additional question after my talk. But now just accept that it can be estimated too. And to express all I said in short words, Klemera and Doubal biological age is just a compromise value best describing observable biomarkers in a sample. That's it. We now understand how Klemera and Doubal works. It's just a compromised value in the compromised value on the one-dimensional curve in the multidimensional space. Okay, Now, a couple of words about results. Why Klemera Doubal is so popular. Currently, it has 305 citations, and most of these citations are from papers where authors try to apply Klemera Doubal method to clinical data, to clinical blood biomarkers, to psychological biomarkers and corresponding clocks called psycho clocks estimators, for example. There are actually many results, many papers using Klemera Doubal approach. But I wanted to emphasize only one paper here. This is a paper from Professor Morgan Levine from 2013, where the Professor Levine showed that the algorithm proposed by Klemera Doubal was able to predict mortality in cross-sectional human data better than more commonly used methods, such as PCA or multiple linear regression, or even penalized multiple linear regression. And this is seen from the table she provided in her paper, where hazard ratio coefficient is the largest among others models. This means that biological age is really a good predictor of mortality, and of course is better predictor of mortality than chronological age. And this, as we discussed before, a good criterion, a criterion of a good biological age estimate. So the Professor Morgan Levine thinks that's a good model. And why Klemera Doubal approach is better than multiple linear regression? This is my thoughts, my experience of working with this particular method allowed me to emphasize not four, but actually three advantages. The first one is, of course, Klemera Doubal allows using chronological age as a predictor within the model, and MLR not, because MLR use chronological age as an outcome, as a target variable. The second, MLR suffers from regression to average phenomenon. To understand this phenomenon, you need to look at this plot, where you see the points that are far from the average, which is equal to 0, are declined from the true value, which is not the case for Klemera Doubal estimations. By the way, this result was proposed in this paper, in this preprint below, which is quite recent and which provides a lot of critics to the aging clocks as a concept. And of course, multiple linear regression suffers from biomarker paradox, which we discussed before. Concerning this paper, I already mentioned this recent bioarchive preprint. I cannot to mention the most important idea from this paper about any aging clocks. This is called, this idea is that any first generation aging clocks you want to train contains within itself an identical association assumption. If you train aging clocks, you already presume that identical association assumption holds. What does it mean? What does it mean, identical association assumption? So, remember the main assumption about biological age, which is the sum of chronological age and acceleration term. Now let's remember the meaningful assumption of Klemera Doubal, that any age-related biomarker can be expressed through the function of biological age and age-independent fluctuation term, or random noise. Next, let's assume that the linear assumption holds that our function f is a linear function of biological age. And let's decompose biological age to the chronological age plus acceleration plus delta term. Here we finally faced with a problem. How do we know that chronological age c and delta determines x with the same coefficient a, which is the slope coefficient here? If you think that chronological age and age acceleration are coupled with x through the same ax, you actually accept identical association assumption in your model. This identical association assumption means that all the features in this plot, each point is a feature, is a biomarker in your data set. And these points are all on the same straight line, because if you use identical association assumption, you use the same a coefficient. Authors also considered 3 other scenarios where identical association assumption doesn't hold. For example, this scenario where inverse association holds. This can be interpreted as that chronological age has positive association with x, and age acceleration has negative association with x, which is quite counterintuitive, but theoretically possible. And two other scenarios are also possible. The first is where there is no identical association assumption, and this means that the feature which for which identical association assumption doesn't hold, actually useless for your aging clocks. And finally, the fourth partial identical association assumption looks like the most the quite real case for such assumptions. The quite not a real case, but it is more similar to the reality. It is more. It seems like that reality works in such way that most of the biomarkers have the same association with x in their sign, but not in their magnitude. It means that x is dependent from the c with slope 1 and from the delta with slope 0.5, for example. It seems like this is the reality, but this is an important assumption about the reality, which does not allow to use any feature you made to imagine or invent. Your feature should satisfy this assumption, at least partially. Okay, I'm moving to the end of our today's session, and I want to discuss the following question. First generation aging clocks. Do we really need them? Do we need aging clocks actually? This question has 2 answers, so obviously yes and no. Why yes? Yes, because they provide simple surrogate measure, potentially utilizable in clinical trials. Yes, because it allows in silico search for new geroprotectors. For example, predicting biological age with reprogramming intervention or other chemical interventions or something, I don't know, heterochronic parabiosis. Yes, because it's just an interesting concept to discuss, of course. But why not? First, no, because it is poorly defined concept and it allows manipulation of results and conclusions, which is quite undesirable for real science. No, because it fails, the aging clock fails, the biological age as a concept fails if identical association assumption doesn't hold, as we discussed previously. And no, because it is one-dimensional measure of health state. Why it is not the best choice to use one-dimensional measure? I propose such an autoencoder analogy because autoencoder tries to compress your information. If you know what is autoencoder, it's just a neural network which tries to decompress information into the latent space like this. And by decompressing, you need a small set of parameters. When you try to compress all information about aging of an organism to the one-dimensional latent space, which also may call biological age, you lose information, and the losses of information can be very high. Instead, you may describe your aging with a set of parameters, with a set of biological age variables. And this would be biological age vector, like this. So instead of using multidimensional latent space, you force it to be one dimension. And this leads to loss of information. In summary, what do we know today? Biological age is a surrogate measure, potentially useful in clinical trials, but poorly defined and should be used carefully, especially if you especially if you try to invent a new geroprotector with this measure. If you use it, make sure that features you include to the model satisfy identical association assumption. But problem, you cannot check that feature satisfy identical association assumption because it is untestable hypothesis. Because identical association assumption is just an object, is just a hypothesis you may believe or not. But don't worry, in practice, you actually may use features that can at least predict mortality or aging-related disease incidence. And these features are apparently will be a good features for constructing biological age. Model-based or unsupervised approach are better than supervised because supervised approach suffers from different paradoxes, different problems, cannot use chronological age as predictor, and so on. Klemera Doubal is a great example of how biological age can be constructed from meaningful assumptions, meaningful and quite limited set of assumptions. And the last quite pessimistic conclusion that first generation aging clocks are quite outdated. If you would ask me, I wouldn't try to fit new first generation aging clocks, but rather I would try to gather more data, and it would be better to train second generation aging clocks. But maybe next time about them. Thank you very much, guys. Thank you that sustain this presentation, which is quite long today. Thank you.
Thanks, Dmitrii.
Questions now. Questions, guys. Questions, discussions. I really wonder about what do you think about all this stuff. Let's discuss some moments.
So I suppose this is a bit more in the realm of like the application in the lab of running these things. But do they tend to take samples like kind of from different organs? Because I'm bearing in mind like there's the growing body of literature saying that there's different aging rates in different organs, and so I'm wondering if different organs might give different kind of results on the biomarkers.
I agree, definitely I agree that different organs age with different rate. It in principle one brilliant example is that our brain tissue ages much slower than, for example, our skin. I noted this from a ton of different data, even from the chromatin data like ATAC-seq, like HiC, like transcriptomics. Brain suffer from aging much less than skin, than liver, than blood, even blood, for example. The important thing here, however, that if we try to measure biological age in different organs, is the as a rule, we use transcriptomics data, for example, or biomarkers data, for example, like blood biomarkers, or if these biomarkers are actually correlated with mortality, we don't know. If this biomarker correlated with age-related disease, we don't know. If yes, then probably they are good predictors for good features for biological age. Yes. So let me return to the Zoom. I want to see you guys. Yeah. If the presentation will be neat, I return to the slides and turn on the turn on them again.
When you say about blood biomarkers, what kind of biomarkers is it generally that they use for aging clocks in blood? What kind of features?
One of the most citable the biomarker is an albumin level, because it has a strong correlation with age. This is a red blood cell red cells count, white lymphocytes count, hematocritis. This is all these biomarkers you actually can find in dataset that is called NHANES. For example, this dataset contains 22 biomarkers, if I'm not mistaken, and all these biomarkers are from blood samples. But albumin, I believe, the most important in terms of correlation. A question from the chat. I haven't heard about the second generation bioclocks exactly, because I didn't mention them. Are they distinguished from first generation by the data or markers collected, or by the use of different, perhaps nonlinear mathematical techniques model? You're absolutely right. This is brilliant question, because second generation aging clocks much more I would say correct way to construct biological age predictor, because second generation aging clocks main difference is that second generation aging clocks use mortality directly, mortality measure, which is measurable. We can measure mortality because we know the date of the death for a particular individual. And these second generation aging clocks use mortality as an outcome which we want to predict. And since we use it as an outcome, we may select features that are directly correlated not with some spurious chronological age, but with mortality. And this is the loop is gathered because we have a good outcome which can be measured, which is clearly defined, and we have features that can be used to predict this outcome. So the second generation aging clocks are brilliant, and I believe that the future is for second generation clocks. Yes. Any other questions, guys?
Hello.
Anya,
I'm,
hello.
I have a question. Yeah.
Mhm.
I didn't understand this individual association assumption.
Mhm.
Could you repeat,
Yes.
please?
Suppose you have true biological age. As we said in the presentation, the biological age can be decomposed to the chronological age and age acceleration terms. And you have some biomarker, for example, albumin level. You know that albumin level correlates with chronological age. For example, the coefficient of correlation 0.5. But we have age acceleration term. Age acceleration term can be coupled with lifestyle of an individual. For example, for smoking people, age acceleration will be higher. For healthy people, age acceleration will be lower. Obviously, that albumin level may correlate with age acceleration in separate way than its chronological age does. For example, let age acceleration correlates with biological age with coefficient 0.25. But now look, we have different coefficients of correlations between biological age, true biological age, and age acceleration and chronological age. And this means that our associations are not identical. Our association of biological age with chronological and age acceleration are not identical because different coefficient of correlation, because different slopes in linear regression model. These different slopes violates, if we would train a linear model and obtain different slopes for chronological age and age acceleration, we violate identical association assumption. Is it clear now?
Uh-huh.
Great. Thank you. Thank you for the question, Anya. So, I believe Evgeny wants to ask me something because I'm kind of attacking his master thesis.
Well, yeah, hello everyone. I guess I
Hello.
can be heard through some noise. But yeah, well, I still think that, well, probably the most attacking point is this identical association assumption. But
Exactly.
I still think that if we can actually predict anything, if we can actually predict chronological age with some accuracy, then the biomarkers that we use to build our predictions upon are probably kind of they kind of hold up to this assumption already, if we can make any predictions from them. So I don't think that this point, this is a valid point, but it doesn't destroy all the talks altogether, because if they are able to predict at least something, and probably
Uh,
uh,
yeah, I have a problem with connection, Evgeny.
probably
You yes, probably maybe if your question is short, maybe you will ask it in the chat.
yeah. Well, the question, it's not like the question. It's more like
Comment.
yeah. I'll just ask it in the chat.
Okay.
I have a quick question. Well, then, if possible.
Sure.
Hi, Dmitrii. Hi, everyone.
Hello.
Thank you a lot for
Hello.
the talk.
Happy to see you here today.
I really want to know if the second secondary biological clocks
Second generation.
Second generation of clocks, if they are all about going from one particular variable of biological age to a vector, because I feel like it increases the space. It increases the space of how you can adjust to personal parameters and every situation, but it reduces the interpretability for me, like as it feels. What do you think about it? Yes.
This is a nice comment. Exactly. If we increase the dimensionality of our health measure, it decreases interpretability definitely. Second, I should say that second generation aging clocks not about multidimensionality. Second generation aging clocks about coupling between biomarkers and mortality. But this is a separate story. This is a separate story. And multidimensionality of biological age is also a separate story. And of course, you're absolutely right that multidimensionality decreases the interpretability, but it may increase, in principle, efficacy of the measure. Because if we measure aging, biological aging, as a multidimensional measure, we could catch aspects of aging. Because one aged human may not be similar to another aged human.
Absolutely.
And if we learn how to account this, our geroprotectors, our clinical trials would be more right, more clear. So I think that multidimensional aging measure could be useful in some situations, but in the situations where you need only one number, of course, you may return to the one-dimensional biological age estimators.
Got it. Thank you. We'll be happy to listen about the second generation clocks more.
I believe for this story, a separate session will be needed. But if you're interested in this, I especially recommend you to Google PhenoAge. I will text this. PhenoAge. PhenoAge is a starting point for digging into the world of second generation clocks. It is also a paper from Morgan Levine group. This is quite a clever approach, and it uses mortality, of course. So
thank you. Thanks.
You're welcome. The question from the chat from Evgeny, doesn't it mean that if we select some biomarkers to predict chronological age from, and the predictions are quite accurate, then the identical association assumption is kind of embedded into these biomarker already? Whoa, tricky question. Let me read it one more time. Doesn't mean that if we select some biomarkers to predict chronological age, and the predictions are quite accurate, then an identical association assumption. Short answer, no, it doesn't. Because your biomarker can be really well correlated with chronological age, but in principle, it cannot answer the question about biological health organism status. And the simple example, let me give a simple example here. We know that the color of hairs is correlated with age, but early whitening colors, I forgot the word for describing the whitening process, or let me check.
No.
Sorry? Yes,
Gray-haired?
gray-haired. Yes, gray-haired. We know that simple fact that gray hairs are not very well correlated with health status, but it is correlated with age. You may obtain gray hairs quite early, for example, in 30 years, in 40 years, but you will be absolutely in the good physical form. And this is an example of biomarker, gray hairs, which isn't correlated with age acceleration, but it is correlated with chronological age. Another example is weight, by the way. Weight of the human. Weight is correlated with chronological age, of course. And in the later ages, you start to lose weight, but you will not be more healthier from this. So there are many biomarkers which are correlated with chronological age. For example, income is a good biomarker. Is a good correlate. Is a good has a good correlation with chronological age. Your income, basically, but it does not reflect your biological age status, your healthy status. So this is a bad biomarker. Your income is a bad biomarker. Though your income may provide you possibilities to be more healthy, so maybe this correlation is not so straightforward. Okay.
So you said that you wouldn't try and fit new data to one of these first generation clocks, and that you would be more likely to go for a later generation clock, right?
Yes. When I say that I wouldn't try, I mean that I wouldn't try to invent new model predicting chronological age from set of biomarkers. For example, definitely I will not train neural networks, deep neural transformers with ton of embeddings on biological biomarkers to predict chronological age, because it's quite hype. It's quite of a hype, but it's useless. It's quite useless for the progress in the aging domain. For the progress in aging domain, I would try to train hazard models. These are the models which use mortality as an outcome. They are semi-parametric, and survival analysis, by the way, in our computational biology of aging book, we discussed shortly these models.
Like a GrimAge. Is GrimAge one of those?
Exactly. GrimAge is the second generation model. It's a good paper of 2018 after Morgan Levine, I believe. Yes, it's a very clever predictor. Yes, any model that can predict mortality from biomarkers are much better than first generation clocks. This is my opinion, because this directly falls from my experience of working with first generation clocks, especially from transcriptomic clocks. But I should also add one comment. If your goal not to predict biological age, but chronological age from data, this may be useful. Why? Because if you see that your set of biomarkers has a kind of complex correlation with chronological age, for example, gene expression values, you can really analyze those genes that are correlated with this chronological age. And this correlation is interesting by itself, because if you know the set of genes that are correlated with chronological age, you probably know aging signature. And this aging signature may help you to go to some intervention to understand metabolic pathways of aging, to understand the different confounders in your data and so on. This can be helpful. But if your goal not to predict biological age, but chronological. So, well, so we are already 1 hour in connection. Vitaly, how much time do we have?
Alex, I think if someone have any questions or opinions, we can continue a bit more. But if no, yeah, we can wrap up.
Okay, 1 minute for one additional interesting question. Actually, yes, Evgeny said something in the chats. Yeah. Well, making your gray hair turn colored again would likely count as a rejuvenation. No? That's a good point. I don't know. Some health state underlies graying for sure. Graying is not a sign of some deadly health change, but it's a sign of some change anyways. Nothing to contradict to this, because I agree. Yeah, I agree. Gray hairs is actually is definitely a sign of something, of some changes. This is a right proposition.
I found it interesting when if you've heard of Greg Fahy and his thymus regeneration projects,
which regeneration project?
Greg Fahy has thymus regeneration. I think it was human growth hormone in the thymus to start producing T cells again. He couldn't get yet. There was a point, I can't remember when it was. I watched a talk by him, and he was saying that he was running kind of an ad hoc study with just some of his friends, just to like test the waters a bit. And when he tried it in his friends, the first thing that he saw, and these were all men around his age, they were about 60s, early 70s. He noticed that a couple of them had gray hairs turn back to brown, and that was the first kind of sign that he saw that there might have been some kind of some kind of rejuvenation effect happening there. So I find it interesting that prior to running a biological clock and actually seeing all of these things, it's kind of like the proto-biological clock is is watching the hair.
Unfortunately, I really didn't hear about this project, but it is very interesting that they use color hair for as a biomarker of rejuvenation. It's a super surprising for me, because I previously read and we discussed 1 year ago. We discussed a review paper where authors proved that color of hairs is not it's not a good biomarker of health state, but maybe in their particular framework they decided to use it for somehow. Maybe they considered in particular skin health, and maybe that was the point. So why isn't it a good biomarker of health? Graying is not a sound delta. Let me, Evgeny, let me be more conservative. According to the comment of Dylan, my best guess now that I actually don't know if the graying of hairs is a good biomarker of health or not. So I believe that I need more data. I need more, maybe you know an article where I can read this, where I can find the plot which shows the color of hair and mortality or self-rated aging and so on. And this will be enough to me to believe that this is a good biomarker. Okay, YouTube. Oh, thank you, Dylan. Thank you.
There we go. That's just the bit in he does a TEDx talk. And I think it's just while he's waiting for the results to come in, right? Like a more definitive results. The first thing he notices in his friends is just that the hair darkens a bit. So he just says it's a hopeful sign. It's not nothing. There's nothing scientific. Just a little hopeful.
Okay.